宣华、品兰，好，放下。看一下那个了哈，那个转入和转译的2、3、5、6。2、3，这个。
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is represented.
这个它是5' 到3'，然后这里是5' 到3'，这个要记起来哦，这是5' 往右到3'，这也是5' 往左到3'，这个要记起来。
The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, the 5' end and the 3' end.
方向是5' 到3'。
strand is made continuously. The DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks, Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer, down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
DNA 变成蛋白质
Here
的
is a
过
cell,
程。
the basic unit of all living tissue. In most
细胞
human cells, there is a structure called the nucleus.
细胞
The
核
nucleus contains genome. In humans, the genome is split between 23
孔
pairs of chromosomes.
染色体
Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones.
histone 主蛋白 DNA
Within the DNA are sections called genes. These genes contain the instructions for proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. Types of amino acid.
tRNA
Transfer RNA molecules carry the
那是
amino
氨基
acids
酸，
to the
上面那是氨
ribosome.
基酸，它底下那个是
The messenger
tRNA。
RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid.
你看它氨基酸就一直接在一起，
This is added to a growing chain of amino acids.
然后就
Once the
做出
last
了
amino acid
北
has
境
been。added, the chain folds into a complex 3D shape to form the protein.
Sun. 蛋白质的合成、转译、
Protein synthesis, translation. Inside the body, the process of translation occurs within every single cell. Each cell has a nucleus. After transcription, mRNAs move out of the nucleus and enter the cytoplasm. This mRNA strand acts as a template for protein synthesis. Present in the cytoplasm is an enzyme, aminoacyl tRNA synthetase. The enzyme macromolecule has 2 binding sites. One site recognizes the amino acid methionine. Following the binding of the ATP prophosphate, resulting in activation of amino acid. Finally, tRNA and the activated amino acid bind together. This amino-acylated tRNA is known as met-tRNA and is released from the enzyme. This marks the commencement of the first stage of protein synthesis, the initiation. During the initiation stage, a small ribosome binds to the mRNA strand. Strand is codons are sequences of 3 bases. Then the ribosome subunit moves along mRNA in 5' to 3' direction. It recognizes the AUG codon or the initiation codon. At this point, met-tRNA is possessing the anti-codon AC up with the AUG codon of the mRNA. Then a large subunit of ribosome combines with a small ribosomal subunit. The large subunit shows 3 sites: the aceptor site, or the A site, the peptidyl site, or the P site,
还有3个
the
位置
exit。site, or the E site. This whole unit forms the initiation complex. This is followed by the elongation stage. During this stage, the RNA carrying molecule of an amino acid approaches the mRNA ribosome complex and fits in the A site.
氨基酸来插进去，
Then a bond is formed between methionine and the amino acid molecule on the tRNA. As a result, met-tRNA becomes deacylated. The ribosome then advances a distance of 1 codon. Deacylated tRNA shifts to the E site from where it dissociates. Meanwhile, another tRNA carrying amino acid molecule attaches to the A. This is followed by the binding of the amino acid molecules. Repetition of this process leads to the formation of an amino acid chain. This event is called elongation. Finally, when the UAG codon or the stop codon reaches the A site, elongation is
就
terminated.
停止码。
Termination is the last stage of protein synthesis. The chain of amino acids from the ribosome. This released amino acid chain is the protein. Protein synthesis is known as translation. Then attaches from the mRNA. Ribosome detaches and dissociates large subunits. Summary shows that the first stage involves the binding of the met tRNA to mRNA and the small subunit. The larger subunit of ribosome then combines with the small subunit.
大致单元单位。
The second stage is the elongation stage. In the incoming aminoacyl tRNA fits in the A site. Then a bond is formed between methionine and the acid molecule on the tRNA. Repeated until a chain of amino acid molecules is formed. The last stage of proteins is the termination stage. When the ribosome reaches the stop codon UAG, elongation stops and the formed amino acid chain, which is the protein macromolecule, detaches from the ribosome. Subsequently, ribosomal subunits, along with the tRNA, dissociate from the mRNA.
还有一个6。
Egg proteins are contained in our DNA.
转录啊。
DNA contains genes. A gene, a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. The motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the RNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliciosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can pass through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons.
这就是密码表了。
Most codons code
它
for
的起
specific
始密
amino
码是什么？
acids.
终止密码是什么？
There
然后
are
做出
4
来是什么？
special codons, one that codes for start and 3 that code for stop.
左边是起始码，后面是终止码。好，先到这边吧。等一下贴个那个网址给你们哈，上去练习一下。